Welcome to Curiosity, the podcast where science meets wonder. Join us as we explore the fascinating, the innovative, and the unexpected, from the secrets of the cosmos to the marvels of everyday life. Each episode takes you on a journey to uncover the knowledge and stories that fuel our endless quest for understanding. Let your curiosity guide you.
You know, there is this really romantic image we all tend to have of agriculture. You picture a rolling green field, a farmer working the land, the sun setting, and you think to yourself, this is nature. You walk into the produce section of your local grocery store, you pick up an ear of corn or a banana, and you just assume you are holding something that the earth basically provided as is.
Yeah, it's a beautiful image. It is also, historically speaking, completely fabricated.
Right. It's a myth we tell ourselves, because if you actually hopped in a time machine and went back, say, 10,000 years, you wouldn't even recognize the ingredients for your salad.
Not at all. I mean, natural corn, for instance, it was a wild grass called teosinte. It had these tiny, incredibly hard kernels that would probably break your teeth if you tried to eat them. The natural ancestor of the banana was full of hard seeds and was practically inedible. The reality is that agriculture, by its very definition, is an act of genetic intervention. And it has been for 10 millennia.
So in a sense, we've been modifying organisms since the Neolithic Revolution. We didn't call it genetic engineering back then. We just called it farming.
Exactly. We called it selection, crossing, breeding. But the intention was always the exact same: to manipulate the genome. We wanted higher yields. We wanted better flavor. We wanted the plant to survive a sudden cold snap. The crops that form the absolute bedrock of our modern food system, hybrid maize, dwarf wheat, disease-resistant vegetables, these are not wild plants. They are the cumulative result of centuries of very deliberate human manipulation.
But, and this is the crux of what we are covering in this deep dive today, there's a massive difference between how we did it back then and how we are doing it right now. The sources we have for this deep dive are incredibly dense. They cover everything from molecular biology to global trade law. But the central theme, the absolute core of our mission today, is exploring this massive shift in precision.
Precision is definitely the key word there. Traditional breeding is effectively a sledgehammer. When you cross two plants, you are shuffling entire chromosomes. It is a highly stochastic process.
Stochastic, meaning like random.
Yeah, meaning it relies heavily on chance and statistics. You are mixing tens of thousands of genes, hoping to get one specific trait, maybe a red apple, but you invariably drag along a vast amount of uncharacterized genetic material with it. You might get the red apple, but you might also accidentally drag along a gene that makes the fruit bitter or makes the tree highly susceptible to a fungus. It's really messy.
It's like trying to edit a book by just, shuffling the chapters around.
Yeah.
You might get the ending you want, but you've probably completely ruined the character development in chapter 3.
That is a perfect analogy. And that brings us to the real paradigm shift. The exact moment where we stopped shuffling chapters and started editing individual words.
Pierre talking about CRISPR here.
We are. Specifically, the development of the CRISPR-Cas9 system, which was first described in its programmable form back in 2012 by Jennifer Deogna, Emmanuel Charpentier, and their colleagues. This wasn't just a new tool for the toolbox. It was a fundamental disruption in plant science. We fully transitioned from macroscopic manipulation, looking at the plant and hoping for the best, to nucleotide-level molecular precision.
So no more crossing your fingers and hoping the right genes matched up in the offspring. This is literally find and replace for DNA.
Precisely. And for us to really unpack that today, we need to understand the molecular mechanism. How it actually works under the hood dictates why it is so incredibly powerful. Then we'll look at the specific applications, from fighting climate change to saving the banana from extinction. And finally, we have to wade into the global regulatory swamp, because the politics governing this are just as complex as the biology.
Let's start with the biology then. When I was reading through the technical breakdowns in our sources, one thing really stood out to me. We talk about CRISPR as this futuristic, cutting-edge technology, but it's actually ancient. It's not something humans invented from scratch in a lab. It's something we found in nature.
That is the great irony of biotechnology. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. It actually originates as an adaptive immune system in bacteria and archaea.
Wait, let's pause right there. Bacteria have immune systems. Because I think most people listening probably assume bacteria are the infection. They don't think about bacteria getting infections themselves.
Oh, it is a brutal, microscopic war zone down there. Bacteria are constantly under attack by viruses called bacteriophages. So over millions of years of evolution, bacteria developed a defense mechanism. When a virus attacks, if the bacteria survives, it keeps a little snippet of the attacking virus's DNA.
Like a biological trophy.
Yeah. Kind of like a mugshot. It stores that little snippet of viral DNA in its own genome, specifically in the CRISPR array. If that exact virus ever shows up again, the bacteria pulls out the mugshot, recognizes the intruder immediately, and destroys it.
So Doudna and Charpentier essentially looked at this incredibly efficient bacterial defense system and said, wait, if bacteria can program this system to chop up viral DNA, why can't we just program it to chop up any DNA we want?
Exactly. They repurposed a bacterial shield into a universal pair of molecular scissors. And the whole system relies on two primary components. If you want to understand modern gene editing, you have to understand these two players. First, you have the guide RNA, or gRNA.
This is the GPS of the system, right?
Correct. The guide RNA is a specific string of nucleotides genetic code that exactly matches the target you want to hit. If you want to edit a specific gene in a wheat plant that causes it to be susceptible to mildew, you program the guide RNA with that exact genetic address.
And the second component is the actual muscle that does the work.
Right, the Cas9 protein. This is an endonuclease. Its only job in the world is to cut DNA. The guide RNA grabs the Cas9 protein, drags it to the specific location in the genome, and Cas9 induces what we call a double-strand break. It snaps the DNA double helix cleanly in half.
Okay, but here's where I get a bit stuck. Snapping DNA sounds bad. If I snap my leg bone, I haven't exactly improved my walking ability. How does breaking the DNA lead to a better, stronger crop?
That is the most brilliant part of the whole process. The magic isn't actually in the cut itself. The magic is in the repair. As soon as Cas9 breaks the DNA, the plant cell panics. Its endogenous repair machinery rushes to the site to fix the massive breach. We are essentially hacking the plant's own emergency response system. And the final outcome depends entirely on which repair pathway the cell decides to use.
The sources mention two distinct pathways for this, NHEJ and HER. Let's break those down, because the distinction seems to matter a whole lot for what you can actually achieve agriculturally.
It matters immensely. The first one, non-homologous end joining, or NHEJ, is the quick and dirty method. The cell just grabs the two broken ends of the DNA strand and frantically glues them back together.
With no template, no instructions.
Just emergency triage. And because the cell is rushing to fix the break, it is an error-prone process. The cell often accidentally adds a random genetic letter or deletes a few letters right at the join site. We call these indels, which is short for insertions or deletions.
But in this specific context, an error is actually a good thing.
If your goal is destruction, yes. These small errors usually cause a frameshift mutation. They disrupt the coding sequence of the gene. The gene effectively becomes gibberish, and the cell just stops using it entirely. We call this a knockout.
So if there's a gene that makes a plant weak or susceptible to a specific bug, you use this quick and dirty method to just break that gene permanently.
Exactly. You silence the vulnerability, but sometimes you want to do a lot more than just break things. You want to rewrite them. That is where the second mechanism comes in homology-directed repair, or HDR.
This is the high-precision method.
Right. In this scenario, you don't just put in the CRISPR scissors. You also provide a repair template. It is a piece of DNA that looks almost exactly like the broken area, but it contains the very specific change or substitution you want to make.
So the plant cell sees the break, looks around, sees this template lying there, and thinks, oh, here are the original blueprints, and uses it to fix the damage.
That is exactly what happens. It copies your edited sequence seamlessly into its own genome. This allows us to substitute specific DNA sequences or insert entirely new traits with unbelievable precision.
That sounds incredibly elegant on paper. But practically speaking, getting these molecular tools into a plant cell sounds like a total nightmare. Plant cells aren't like animal cells. They have that thick, rigid cell wall. It's like trying to perform delicate microscopic surgery inside a heavily armored fortress.
It really is a fortress, and the technical methods we use to breach it are, quite frankly, pretty sci-fi. One of the most common methods for plant transformation is called ballistic delivery, but everyone in the lab just calls it the gene gun.
Please tell me that is literally what it sounds like.
It is. We take microscopic particles of gold or tungsten. We coat them with the DNA encoding the CRISPR components. We load them into a pressurized device, and we literally fire them at the plant tissue at high velocity.
You were shooting gold bullets at pieces of corn.
Essentially, yes. We physically punch right through the cell wall. It is a brute force method, but it works surprisingly well. The other primary method is a bit more biological. We use a Trojan horse. There is a soil bacterium called Agrobacterium tumefaciens.
I recall reading about this. In nature, this specific bacteria attacks plants by injecting its own DNA into them to cause these big tumors, right?
Correct. It is a natural genetic engineer. So scientists simply disarm it. We strip out the genes that cause the disease and the tumors, and we replace them with the CRISPR machinery. We then let the bacteria infect the plant just like it naturally would. It acts as the delivery vector, dropping our editing tools directly into the plant cell nucleus.
That is ingenious. Using a pathogenic bacteria to deliver a bacterial immune system into a plant cell. It really highlights how interconnected all this biology is. But before we move to the crops themselves, we have to clear up the biggest point of confusion for the public, the GMO label. To the average person listening, this all just sounds like GMOs. But the scientific community is adamant that CRISPR is fundamentally different. Why is that?
This distinction is absolutely critical, both biologically and legally. Traditional GMOs, transgenics, involve the integration of foreign DNA. That means taking a gene from a completely different species, like taking a gene from a bacteria or a fish, and forcing it into a plant's genome.
That's the whole Frankenfood fear, crossing the natural species barrier.
Right. But many CRISPR applications are what we call cisgenic. We are not introducing any foreign DNA at all. We are modifying the organism's existing genome. We are turning a gene off or tweaking a promoter that is already native to the plant. The end result is often genetically indistinguishable from a random mutation that could have happened naturally in a field, given enough time and luck.
And that biological distinction completely changes the economics of the whole agricultural industry, doesn't it?
Completely. Developing a first-generation transgenic GMO is incredibly expensive. We're talking hundreds of millions of dollars and a decade or more of regulatory hurdles. That cost structure meant that only massive global corporations could afford to play the game, and they only focused on huge commodity crops like corn, soy, and cotton.
Because if you are spending $150 million on research and compliance, you need a massive blockbuster product to recoup that. You aren't going to spend that kind of money to slightly improve, say, lettuce or cassava.
Exactly. But CRISPR totally changes the equation. It is exponentially cheaper, faster, and more precise. If regulators accept that cisgenic edits aren't traditional GMOs, the cost of development plummets. Suddenly, public researchers and small startups can justify editing minor crops, specialty vegetables, and crops that are absolutely critical for food security in the developing world. It democratizes the technology.
So the tool is sharper, it's faster, and it's cheaper. Now let's talk about the actual problems we are trying to solve with it, because the clock is definitely ticking. The sources make it very clear the global climate is changing rapidly, and our current crops are simply not built for the world to 2050.
This is what we call the climate imperative. We are facing radically shifting precipitation patterns, rising average temperatures, and increased soil salinity. Plants are stationary organisms. They can't just migrate to cooler climates when things get tough. They have to adapt or they die. And natural evolution is simply too slow to keep up with the current rate of anthropogenic climate change.
Let's look at the water issue first. Drought. What are scientists actually editing to help a plant survive without water?
One of the most fascinating areas of research is the regulation of stomata. These are microscopic pores on the surface of the plant's leaves. Think of them as the plant's mouths. They have to open to let carbon dioxide in so the plant can perform photosynthesis.
But every time they open their mouths, water escapes into the air.
Yes, it is a fundamental physiological trade-off called transpiration. If you open wide to eat, you dehydrate. Researchers are now using CRISPR to edit the signaling genes that control the aperture of these pores. They are optimizing the balance, teaching the plant to close the pores slightly more or close them much faster in response to dry air.
So you get a plant that is much more water efficient without actually starving it of the carbon dioxide it needs to grow.
Exactly. We have already seen significant experimental success with this stomatal regulation in rice, wheat, and maize. But we are also looking underground. Root architecture is just as critical for drought survival.
Which makes intuitive sense. If the topsoil is dry, the plant needs to go deeper to find water.
Right. But traditional agricultural crops often have very shallow root systems. CRISPR is currently being used to modify the specific genes that determine root depth and root density. In maize, for example, they've created experimental lines with roots that grow at a much steeper angle, punching deeper into the soil profile to access moisture reserves that standard plants simply can't reach.
It's like upgrading the plant from a short straw to a deep vacuum pump. But then there's the other massive water problem mentioned in the sources: salt. Salinity is highlighted as a major threat, which might actually surprise people who don't live in arid, irrigated regions.
It is a silent killer of global farmland. When you irrigate in hot climates, the water evaporates quickly, but the trace salts in the water stay behind in the dirt. Over decades of farming, the soil becomes increasingly salty. It creates a highly toxic osmotic environment where most conventional crops simply wither and die.
So how do you use molecular scissors to teach a plant to survive in poison?
You edit the plant's bouncers. Plants have specific genes that control transport the proteins that move minerals in and out of the cells. Scientists are editing these transporters to either exclude sodium at the root surface, basically not letting the salt inside the plant at all, or if the salt does get in, to actively sequester it.
Sequester it where?
In the vacuole. It is a large cellular storage compartment inside the plant cell. The edited plant effectively locks the toxic sodium away in a biological safe room where it can't interfere with the delicate metabolic machinery of the rest of the cell. This specific edit could allow us to grow food on millions of acres of land globally that are currently considered entirely marginal or useless.
That is incredible. It's not just about protecting the current yield we have. It's about physically expanding where on Earth we can actually farm. Now, moving from the harsh physical environment to the biological enemies. Biotic stress, the bugs, the fungus, the relentless bacteria.
This is the eternal war of agriculture, the evolutionary arms race. We breed a resistant crop. The pathogen evolves to overcome it a few years later. We breed a new one. The pathogen evolves again. It never stops.
And the sources highlight a very specific case study on wheat powdery mildew from the Chinese Academy of Sciences. This seemed like a massive technical achievement in the literature. Why was this specific project so incredibly difficult to pull off?
Because wheat is an absolute genetic monster. Humans are diploid. We have 2 sets of chromosomes. Wheat is hexaploid. It has 3 entirely complete genomes labeled A, B, and D, completely mashed together in one plant.
So every single gene in wheat exists in 3 distinct copies.
Exactly. These are called homoalleles. Now, imagine you want to disable a specific gene that makes the wheat susceptible to powdery mildew. If you try to use traditional breeding to mutate one copy,
the other 2 copies are still sitting there doing the job perfectly fine.
Exactly. The disease still gets in.
Yeah.
It is practically impossible to mutate all 3 copies simultaneously using conventional breeding without ruining the rest of the plant's genome. But CRISPR, it is a guided missile. You program the guide RNA to target that specific genetic sequence, and it goes into the hexaploid genome and cleanly cuts all 3 copies, A, B, and D, at the exact same time.
And the practical result of that?
A wheat plant with durable, robust resistance to powdery mildew with absolutely no penalty to the crop yield. It is something that was theoretically possible, but practically impossible before precision gene editing.
You mentioned susceptibility genes there. That concept really struck me while going through the sources. Usually when we think of giving a plant disease resistance, we think of giving the plant a new shield or a new chemical weapon. But here, we're actually taking something away.
It is a profound philosophical shift in plant pathology. Pathogens don't just brute force their way in. They often hijack the plant's own native proteins to infect it. They need a welcome mat, or a very specific molecular handle to grab onto. By using CRISPR to knock out those specific host genes, the susceptibility genes, we are essentially hiding the doormat.
So the pathogen arrives at the cell, but it literally can't find the handle to open the door.
Precisely. This strategy is currently being used in rice against bacterial blight caused by Xanthomonas oryzae. It is potentially much more durable than adding a foreign resistance gene because the pathogen has to completely reinvent its entire mechanism of attack rather than just mutating slightly to overcome a single introduced toxin.
Okay, we absolutely have to talk about the banana, specifically the Cavendish banana. This is probably the most relatable example for anyone listening because everyone eats them and they are all completely identical.
The Cavendish is the ultimate poster child for agricultural vulnerability. It is a global monoculture. Every single Cavendish banana you see in every supermarket worldwide is a genetic clone of the exact same plant.
Which means if a disease figures out how to kill just one of them?
It can kill every single one of them. And that is exactly what is happening right now with the TR4 strain of Fusarium wilt, which is also known as Panama disease. It is slowly marching across the globe, wiping out plantations, and the Cavendish has a fatal biological flaw. It is completely sterile.
Meaning you can't breed it with anything else.
No seeds. No sexual reproduction whatsoever. You cannot cross a vulnerable Cavendish with a wild, disease-resistant banana species to get a better offspring. It is an evolutionary dead end.
So traditional breeding to save the banana is literally impossible.
Correct. Which means targeted gene editing is arguably the only scientifically viable pathway to save the Cavendish. Scientists are currently using CRISPR to insert resistance genes or modify the genome directly to withstand the TR4 fungus. Without this molecular intervention, the commercial banana as we know it today could effectively disappear entirely, just like the gross Michel variety did back in the 1950s.
That really raises the stakes of this whole discussion. It's not just about farming efficiency. It is about preventing the outright extinction of a major global food staple. But let's pivot slightly to the consumer side of things. Saving the banana is great for supply, but what about making our food fundamentally healthier? The sources talk extensively about biofortification.
Yes, and this is where we are actually seeing the very first commercial successes of the technology. The first CRISPR-edited foods to actually hit the commercial market weren't about boosting yield for the farmer. They were about health traits for the consumer, specifically high oleic oils.
This is in soybeans and canola, right?
Correct. Scientists use CRISPR to edit the fatty acid desaturase genes in these plants. This permanently changes the lipid profile of the resulting oil, making it highly stable at very high cooking temperatures.
Which means the food industry doesn't need to artificially hydrogenate it anymore.
Exactly. Industrial hydrogenation creates trans fats, which are terrible for cardiovascular health. By editing the bean itself to produce better oil naturally, we completely eliminate the need for that harmful industrial processing step. It is a direct, measurable public health benefit.
I also saw fascinating mentions of reducing allergens and natural toxins.
Right. In wheat, researchers are actively editing the alpha-gliadin genes to significantly reduce immunoreactivity. Now, it's not a total cure for celiac disease yet, but it could massively help people with general gluten sensitivity. And in potatoes, they are editing the genome to reduce the natural precursors for acrylamide.
Acrylamide is the carcinogen that naturally forms when you fry starchy foods at high temperatures.
Right.
Yes. So a simple CRISPR edit could literally make your French fries safer to eat. But when we shift our focus to the developing world, the goal isn't just healthier frying oil. It's about micronutrients, what public health experts call hidden hunger.
Making basic staple crops fundamentally more nutritious.
Exactly. Increasing essential iron and zinc levels in staple crops like rice and cassava, enhancing provitamin A. For large populations that rely on a single staple crop for the vast majority of their daily calories, these specific micronutrient deficiencies cause severe stunting, childhood blindness, and immune system failure. If you can code the nutrition right into the seed itself, it is a permanent, sustainable solution to widespread malnutrition.
It sounds almost magical when you lay it out like that. But there's one more specific application mentioned in the yield section of our sources that genuinely sounded like science fiction to me. Changing photosynthesis itself. The theoretical shift from C3 to C4.
This is widely considered the holy grail of plant physiology. You see, not all plants photosynthesize the exact same way. Rice and wheat use what we call the C3 pathway, which is relatively inefficient, especially in very hot break conditions. Maize and sugarcane, on the other hand, use the C4 pathway,
Which is like the evolutionary turbocharged version of photosynthesis.
It is a massive evolutionary upgrade. C4 plants are significantly better at converting raw solar energy into usable biomass. The long-term objective, and to be clear, this is a total moonshot project, is to modify the complex regulatory networks of C3 crops like rice so they biologically resemble the highly efficient C4 pathway.
So using CRISPR to literally teach rice to act like corn.
Essentially, yes. We certainly aren't there yet, but even incremental improvements tweaking specific enzymes to be just 5 or 10% more efficient at capturing carbon could lead to absolutely massive jumps in global agricultural yield.
Okay, so we have the technology, we understand the applications, we can potentially save the banana, feed the world, and fight climate change. But now we hit the brick wall. The real world. Politics, intellectual property, and regulation.
The global regulatory landscape for CRISPR is highly fractured. It is essentially a tale of two completely divergent regulatory philosophies.
The United States versus the European Union.
Precisely. The U.S. essentially employs a product-based approach. The USDA looks at the crop and asks, what is the final biological result? If the edited crop does not contain any foreign DNA and it doesn't pose a traditional plant pest risk, they generally do not regulate it as a GMO.
So they basically treat it exactly like a conventionally bred plant.
Yes, and this highly permissive stance has dramatically lowered the barrier to entry. That is exactly why you see so many small startups and university spinoffs in the U.S. working on CRISPR applications right now.
And Europe took the opposite path.
The EU is process-based. In 2018, the European Court of Justice issued a landmark ruling stating that if you use the CRISPR technique at any point, the resulting organism is legally classified as a GMO. Period.
Even if the final genetic result is completely indistinguishable from nature.
It absolutely doesn't matter to the EU framework. They heavily regulate the method of creation, not the final product. This means any CRISPR crop in Europe faces the exact same massive hurdles as first-generation transgenics, years of safety testing, massive compliance costs, and intense political stigma. The global scientific community has been incredibly critical of this ruling, arguing that it completely ignores basic biological reality.
And where does China fit into this global picture? Because they're usually the wild card in biotechnology.
China is currently investing aggressively. They have massive public sector funding pouring into CRISPR research. Their regulatory framework is certainly more permissive than the EU, though still structured, and they have several advanced varieties already moving toward final approval. They clearly see this technology as a critical strategic necessity for their long-term national food security. They are not going to let a philosophical debate slow their deployment down.
The real tragedy here, though, based on the sources, seems to be the impact on developing nations, the countries that arguably need this technology the most for drought resistance. For basic nutrition, they are caught in the crossfire of this regulatory war.
That is what policy experts call the precautionary trap. Many African and Southeast Asian nations look to Europe and the U.S. for regulatory guidance. But much more importantly, they look to them as vital trade partners. If an African nation approves and grows a highly nutritious CRISPR cassava, but the EU strictly bans any imports of it because of their 2018 ruling, the developing nation's economy suffers massively. So they often default to the strictest possible precautionary regulations just to protect their export markets, even if it actively hurts their own domestic food security.
That is a brutal, real-world economic reality. And speaking of economics, who actually owns all of this? Because if CRISPR is the fundamental future of global food, whoever holds the patents is the ultimate kingmaker.
The intellectual property landscape for CRISPR is an absolute minefield. At the foundational level, you have the Braun Institute fighting UC Berkeley over the original core patents. But practically speaking, in agriculture, the major ag biotech corporations Bayer, Corteva, Syngenta have systematically amassed massive overlapping patent portfolios.
Which brings us right back to the consolidation issue of the 90s. If the big corporations own the fundamental tech, they're obviously going to focus on the most profitable crops.
Exactly. There is a massive commercial incentive to use CRISPR to fix minor issues in corn and soy for the wealthy Western market. There is very little commercial incentive for a massive corporation to spend millions fixing millet or sorghum for a subsistence farmer in the Sahel.
So we desperately need a different economic model for those critical food security crops.
We need broad humanitarian licensing. We need well-funded public sector institutions to step in and fill that gap. There are some foundational agreements currently in place to allow royalty-free use of CRISPR technology specifically for humanitarian purposes, but the actual on-the-ground implementation is highly uneven. We have to ensure that this gene revolution doesn't just exclusively benefit the already wealthy nations.
Which leads us perfectly into the ethical dimensions. We really can't finish this deep dive without touching on the moral and ecological implications. The cultural ghost of the original GMO debate is still heavily haunting this new technology, isn't it?
It is deeply ingrained in the public consciousness, but the core arguments are actually shifting slightly. The old playing God argument, the intense fear that we are crossing a sacred species boundary by putting fish genes in tomatoes that is much less relevant with cisgenic CRISPR because we simply aren't mixing species anymore.
So it conceptually feels a bit more natural to the average consumer.
Slightly more natural, yes, but the deep socioeconomic concerns are still very real and valid. The rapid consolidation of corporate power over the food supply, the loss of autonomy for independent farmers, the fundamental right to save seeds year over year. These are the complex battles that will truly define the CRISPR era in agriculture.
And what about the consumer's fundamental right to know? Labeling. If I walk into a grocery store, I want to know if my food has been gene-edited. But based on everything you explained earlier about cisgenics, is a labeling system even scientifically possible to enforce?
That is the great labeling paradox of CRISPR. Consumers heavily demand clear labels. But because precision CRISPR edits can be physically identical to naturally occurring mutations, there is often absolutely no molecular signature left behind. If I hand a lab a CRISPR-edited mushroom and a naturally mutated mushroom, they literally might not be able to tell them apart using any known chemical tests.
So you literally can't test a shipment of grain at a port to see if it's CRISPR-edited?
Not easily, and in many cases, not at all. Enforcement of any labeling law would have to rely entirely on complex paper trails, seed audits, and a bureaucratic honor system, which is a total nightmare for global trade logistics.
Finally, let's look at the broader environment. We talked extensively about the ecological benefits requiring less toxic pesticide, using less synthetic nitrogen fertilizer. But there are always ecological risks when we alter biology, right?
Always. Biology is fundamentally unpredictable. One of the major recognized risks is gene flow. If we create a highly drought-resistant wheat plant and it manages to cross-pollinate with a closely related wild grass weed living nearby,
You might inadvertently pass that superpower to the weed and create a superweed.
exactly, a wild weed that is now highly drought-resistant and much harder for farmers to kill. And then there's the constant risk of pests rapidly evolving resistance to our new edits. If we deploy these CRISPR-enhanced crops globally without enforcing strict stewardship strategies like maintaining non-edited refuge areas for bugs to live in the pests, we'll simply adapt to the new edits, and the billion-dollar technology will fail entirely.
It really seems like we are standing at a massive historical tipping point. The fundamental science is largely proven. The molecular tools are securely in our hands. But the actual future of our food system isn't going to be decided in a sterile laboratory.
No, it won't be. We are rapidly moving out of the era of experimental novelty and entering the era of routine breeding integration. Over the next single decade, CRISPR gene editing will likely just become a standard, everyday part of agricultural breeding. But the global divergence we discussed is incredibly real. We seriously risk creating a bifurcated, two-tier global food system, highly optimized, climate-resilient agriculture for the developed world, and a much slower, philanthropy-dependent struggle in the developing world.
So the final overarching conclusion of our deep dive today is that the ultimate limits of agricultural gene editing are no longer biological at all.
They are entirely political, economic, and value-based. The most important question facing humanity right now is not what CRISPR can do. The biology is proven. The question is what we will actually allow it to do, and crucially, who we will allow it to benefit.
That is a really heavy, vital thought to leave on. We have the ultimate molecular scissors in our hands, but as a global society, we haven't quite decided what picture we're actually trying to cut out yet.
That is exactly it.
This has been an absolutely fascinating deep dive into the biological future of our food. Thank you so much for taking the time to guide us through the genome today.
It was my absolute pleasure.
And to you, listening right now, the next time you peel a standard Cavendish banana, take a brief moment to really appreciate it. It might be a historical collector's item very soon, or if the complex science we discussed today fully succeeds, it might be the vanguard of a brand new agricultural revolution. Thanks for joining us on this deep dive.